Hello everyone, welcome to my channel Life Science Inside Out. If you like my video, please don't forget to like, share and subscribe. In this video, we will talk about antigen antibody reactions. Before knowing antigen and antibody reaction, let's first see what is an antibody or an antigen. An antibody is a large Y-shaped An antibody is a large Y-shaped protein produced mainly by plasma cell, that is plasma B cell, that is used by immune system to neutralize pathogens such as pathogenic bacteria and viruses. The portion of the antibody where the antigen bind is known as paratome. This region and this region, these are paratome. And an antibody usually have two regions. A variable region, the portion where the antigen binds, is called the variable regions because it varies from antibody to antibody and they are specific for particular antigen. But the constant region remain constant, for a particular species or individual, you can say. And there are two chain. One is your heavy chain and the other one is light chain. This is your light chain and this one is heavy chain. This one is light chain. An antigen is a substance that is capable of stimulating an immune response, specifically activating lymphocytes. Suppose if this is your pathogen, or you can say this is the part of a bacterial cell or viral cell, it will have structure like this. These are called antigen. They have different parts like their protein, carbohydrates, lipids can act as antigenic substance that can generate immune response inside the body. Actually, whenever these pathogens enter the body, there will be many immune cells involved which will chop these pathogens into small parts, and these small parts will act as antigen that will be presented by some antigen presenting cell to T cells and B cells. So this antigen, suppose this portion will be called antigen, and suppose this portion binds to antibody, particular antibody, this portion and this portion. These two parts where which is going to bind to the antibody is known as epitope. So what we have learned, the portion of the antibody that binds to antigen is called paratope, and the parts of the antigen which binds to antibody is known as epitope. Antigen and antibody reactions are reversible, non-covalent biochemical reactions, and these are hydrogen bonds, ionic bonds, hydrophobic interactions, van der Waals forces, and ionic bonds. When we talk about antigen-antibody reactions, we should know three things, which includes affinity, avidity, and cross-reactivity. Affinity is the force of attraction between an antigen binding site of one epitope. If this is your antibody, suppose, sorry for the bad drawing. So this corresponds to your affinity. Affinity will be more if the shape of antigen antibody is perfectly similar or same. If not perfect, the force of attraction will be weak. So what is affinity? It's the strength of attraction between strength by which an antigen binds with the antibody. When we talk about avidity, it's the total strength at which an antibody molecule binds to the antigenic determinant or epitope. So we can say as the number of antigen binding site increases, the strength of overall binding also increases. Dissociation of this antigen from antibodies, dissociation of this antigen from antibody will require all the bonds to be broken simultaneously. More force will be required. If we want them to be dissociated from all of this antigen when we compare to this antigen antibody affinity. Okay, when affinity is compared with avidity, more force will be required to dissociate all the antigens. Therefore, in case of a pentamer, avidity will be more but affinity will be less. Okay, because it will require more strength or more energy to dissociate all the antigen from a pentamer than a monomer. Okay, but the affinity of a monomer will be more than a pentamer, because in case of a monomer, the strength at which an antigen binds to an antibody will be more than a pentamer, because pentamers will be loosely attached. So we can say avidity provides more stability to antigen antibody complex. Avidity is of great significance because this stable antigen antibody complex formed will now be eliminated from the cell more efficiently. Now comes the term cross-reactivity. Cross-reactivity means the ability of an antibody to react with similar epitopes on different antigens. That means what? An antibody specific for a particular antigen, suppose, can also bind sometimes to an antigen molecule which have similar epitopes or epitopic region. You can see in this diagram, though this antigen differs from this antigen, but its epitopic region is similar to this antigen. Therefore, the antibody got confused sometime and they bind to different antigens if their epitopic region is somewhat similar. Okay, this is known as cross-reactivity of antigen-antibody reactions. So when we talk about antigen-antibody reaction, this can be in vivo or in vitro. In vivo or in vitro. In vivo reaction means inside the living organism. Inside living organism. And in vitro means in the lab, in the test tube. Okay. In vivo reactions can ultimately lead to elimination, destruction or removal of the antigen, and there can be different types of reaction like neutralization reaction, immunocomplex formation, opsonization, complement system, antibody dependent cell mediated cytotoxicity or ADCC, hypersensitivity reaction, autoimmunity reaction, etc. And in vitro, in case of in vitro, there may be precipitation, immunodiffusion, countercurrent immunoelectrophoresis, agglutination reaction, complement fixation, neutralization, radioimmunoassay, ELISA, fluorescent antibody reaction. All these are included in vitro reaction. The in vitro antigen antibody reaction is used to detect either antigen or antibody, presence of antigen or antibody in a solution or in a mixture of biological sample and concentration of antigen antibody. Whether this is in vitro or in vivo reaction, there are three stages of antigen antibody reaction. Primary stage is interaction between antigen and antibody, but without any visible effects. Okay, we cannot see any effects, but antigen antibody will come to close proximity with each other and try to form weak bonds. Okay. Then secondary stage is demonstrable events. Okay, where your different reactions will occur like precipitation, agglutination, lysis of cell, neutralization, etc. And third stage is your immunity against infectious disease or allergy. So if we talk about in vitro, we actually give emphasis on the secondary stage. so that we can see the visible reaction, what are the changes occur, whether precipitation occurred or agglutination occur. So we just take into consideration this secondary stage. In this video, we will talk about in vivo immunological reactions. So in vivo reaction, there are different types of in vivo reactions, as I've already told. First is your neutralization reaction. Neutralization reaction can be of two types: toxin neutralization and viral neutralization. In case of toxin neutralization, neutralizing antibody, these are neutralizing antibody that are capable of binding to toxins produced by various pathogenic organisms. Okay, these antibodies are called as antitoxin or neutralizing antibody. What happens whenever there is a toxic molecule that is supposed to bind to this susceptible cell, antibodies like this neutralizing antibodies will come and bind to the toxin molecule and therefore neutralize them, and they are not able to bind to the target cell or susceptible cell, so the cell remain undamaged here. Okay. Second type is your viral neutralization process. And then neutralize the viral infections. Okay, because whenever this antibody bind to this virus molecules, this virus molecule becomes inactive. However, fixation of the classical pathway, especially the complement component C3b to the virus, also aids to viral neutralization process. We will discuss about complement system later in this video. So this is how neutralization reaction occurs. Second is your immunocomplex formation. Antibody possess at least two antigen binding sites. These two are antigen binding sites, and most antigen have at least two epitopes. Okay, most of the antigen have two epitopes or antigenic determinants. The antibody cross-link antigen forming large aggregates of antibody and antigen referred to as immunocomplex. This is known as immunocomplex, which are more readily phagocytosed than our free antigen. So whenever antibody form immunocomplex with antigen structure like this, they are more readily phagocytosed by various phagocytic cells rather than antigens moving freely in the blood plasma. Okay. And then this precipitation or immunocomplex reaction can be of two types. Number one, precipitation reaction. Whenever an antibody binds with a soluble antigen, it forms precipitation. And number two is agglutination reaction. An insoluble antigen or surface bound antigen when bind to antibody, then it forms agglutination reaction. Actually, we will discuss about precipitation and agglutination in vitro reaction more clearly. In opsonization, what happens? Phagocytic cells, they used a complement system for phagocytosis, to enhance the phagocytosis mechanism. There are various proteins in complement system like C1, C2, C3, C4, many more. And this what happens, this is suppose your target cell. This is a cell which you need to destroy. C3b protein will bind to their surface here. Okay, these are C3b protein. These are C3b proteins. Okay. Now what will happen? Antibodies will bind to the target molecule because antibodies will have specific will have specificity for the antigenic determinants in this target cells. And secondly, this phagocytic cells can bind to this Fc portion of antibody because they have Fc receptor. These antibodies then direct binding of this phagocytic cell to the C3b um, complements attached to these cells, because phagocytic cells also contain receptors for C3b proteins. So now they can bind more firmly to this target cells and then engulf these target cells by the method of phagocytosis and now kill the cell. This process is known as opsonization. So here we can see, suppose this is your target cell and we need to eliminate this cell. What will happen? Many cytotoxic cells, these are called cytotoxic cell, NK cell, eosinophil, macrophages, because they produce some cytotoxic substances like your lysozyme, then perforin. All these substances are able to kill the target cell. So this cytotoxic cell have cytotoxic potential express membrane receptor for the Fc region of antibodies, antibody molecule. When antibody is specifically bound to this target cells, whenever there will be binding of the antibody molecule, what happens? The cytotoxic cells can bind to these target cells with the help of this antibody, because they have receptor for antibody molecule. Okay, this Fc portion can bind to the receptor of the cytotoxic cells and subsequently cause the lysis of the target cells. If we consider it as a virus or bacteria, it will have some antigenic determinant that will be specific for some antibodies, and antibodies will come and bind to this antigenic determinant that will help this cytotoxic cells to bind to this antibody. They will indirectly bind to this cell by binding to this antibody molecule. Although the cytotoxic molecules are so even if they are non-specific for this antigen, because cytotoxic molecules we know are not specific, okay, so even though they are not specific, but they have receptors for antibody molecules, so antibodies are ultimately specific for this antigen molecule, so this way they will easily can bind to this target cells. This type of antibody mediated killing of a target cell is known as antibody mediated cytotoxicity, or ADCC in short forms. The cells that can mediate ADCC are NK cells, macrophages, then monocytes, neutrophils, eosinophils, etc. Killing of helminthes is an example which involves antibody mediated killing by eosinophil molecule. Whenever this cytotoxic cell bind to the target cell, they become more active metabolically, and their cytoplasmic content increases, which include lysosomic substances, or you can say granular substances. They release some granular substances, okay, that leads to the lysis of the cells. In addition to this, they also secrete some tumor necrosis factor, or TNF, which may have a cytotoxic effect on the bound target cells. NK cells and eosinophils are also known to secrete some substances called perforin, which are also involved in perforin mediated membrane damage. Hypersensitivity is another reaction. It's an antibody reaction that you can say it's an allergic condition that occurs due to increased immune sensitivity, in which the body reacts to an antigen with an exaggerated immune response. Exaggerated immune response, or you can say allergic reaction. It's usually harmful, okay, for the individual because of tissue damage. This type of hypersensitivity reaction can be manifested in an individual on second or subsequent contact with an antigen and appears immediate or delayed. Number 7 is your autoimmunity. It's a condition characterized by the presence of autoantibody and self-reactive lymphocytes or T cells. Autoimmunity manifests when the body loses immune tolerance. That means what? Body is unable to distinguish between itself and non-self antigenic molecules. Okay, they will not attack the foreign antigen. Foreign antigen don't attack, but instead they will mount an abnormal immune attack either with the antibody or T cell against a person's own self antigen. So we have learned about various antigen-antibody reaction inside a living organism, which we know as immunological reactions. The reaction that occurs in the test tube, or you can say in the lab, are known as serological reactions, and we will discuss about serological reaction in my next video. Thank you for watching.